Okay, in the last video recording we had sort of those introductory slides on structural divisions of the nervous system and and functional divisions of the nervous system, as well as of neurons. And so it's kind of a review of information that we've talked about before. In this video recording we'll be talking about glial cells in more detail than we have before. We've talked about two types of glial cells, neurolemmacytes and oligodendrocytes, but this time we'll be adding four more. Let's see, we'll start with some basic characteristics. Glial cells are, an older term here is neuroglia. Glial cells can be found in the central nervous system and in the peripheral nervous system. These are cells that have a supportive function. They're smaller than neurons. They are able to divide and reproduce and replace themselves essentially. They are non-excitable, so they do not transmit nerve impulses, unlike neurons. And there are far more glial cells than there are neurons, about up to 1 trillion glial cells in comparison to about up to 100 billion neurons. So we do continually make or reproduce new glial cells to support those long-living neurons that we have. So in a generalized summary here, we can see that glial cells are responsible for supporting neurons, both physically to protect them and to help nourish them, depending on the glial cells we're talking about, and to also find a structural framework or scaffolding onto which other nervous system cells are organized or are found. All right, so we'll take a look here at the first type of glial cell, which are astrocytes, and these are notes that you have in your outline, so we'll use the diagram as we talk about the different characteristics of astrocytes. So astrocytes have this sort of aster shape or star-like shape. They're the green cells that we're looking at here. You can see there's a nucleus and multiple processes extending out to other cells here. So these are the most abundant of the glial cells. Over 90% in some areas of the brain is made up of the tissue is made up of glial cells. So some of the various functions that astrocytes help carry out, some of the various functions or roles that they play, one of which is to help form the blood-brain barrier. The blood-brain barrier is, that's sort of demonstrated here, is a feature in the brain that restricts or controls what the materials that are allowed to cross over the cell membranes of endothelia and out into nervous tissues from capillaries. So basically this nervous tissue is very sensitive to toxins, for example, or the interstitial fluids that have the right or don't have an optimal ion concentration. So the blood-brain barrier limits what can enter brain tissue, basically. So the parts of the astrocyte that you can see here, these are called perivascular feet that sort of adhere or attach to capillaries, sort of blocking what can pass through the capillaries. And capillaries tend to, these capillaries tend to be specialized in the brain to limit passage of materials across the membranes here to enter the nervous tissue as well. So we'll talk more about the blood-brain barrier when we get into the brain. But that's one of the functions astrocytes help carry out to protect the brain tissue. They also, in addition to limiting what can enter the brain tissue from capillaries, they help regulate fluid composition. So that refers to the ion composition of the interstitial fluid that's surrounding brain tissue coming from capillaries. The astrocytes also form a structural framework in nervous tissue in the central nervous system. We only have astrocytes in the central nervous system. So you can see how it's sort of supporting multiple cells here and tissues, kind of holding them in place. So this is a sort of scaffolding or structural framework for the nervous tissue. Astrocytes, when neurons are damaged, will replace neurons, fill the spaces in which the neurons were found, when neurons are damaged or die. So it's like a space filler. And they also assist in neuron development in the fetus. They secrete chemicals that help regulate the connections between neurons. So astrocytes have a number of functions here. If we go to the histological view, we can see some examples shown here in tissue showing astrocytes. So they do, especially down here, have that aster-like appearance. Here we're actually looking at the blood-brain barrier. So you can see some perivascular feet attached to a capillary here. All right. Ependymal cells are another type of glial cell found in the central nervous system, like astrocytes, but carrying out a different function. So these tend to be cuboidal epithelial cells that are ciliated, meaning they have sort of hair-like appendages extending from their apical surfaces, and they're found in the brain and spinal cord, the central canal of the spinal cord. And their importance here is associated with this term, choroid plexuses. So they help form choroid plexuses with capillaries in the central nervous system, especially specifically in the brain, to produce cerebrospinal fluid, and that's what CSF stands for. We'll see microglia here. They're basically cells that phagocytize cellular debris, and then there are oligodendrocytes, which we've talked about before, that form myelin sheaths around axons. So we're looking at well, let's do ependymal cells first. We're looking at ependymal cells here. So just to give you a reference of this area of the brain, remember this whole square here is a blow up of this region of the brain, and we're looking at a frontal or coronal section through the brain, slicing the brain so that there's an anterior and a posterior portion. And this blue area highlights a ventricle, which is a chamber in the brain that we haven't talked about yet. But in this chamber, there, right here in the roof of this particular ventricle, is what we're looking at right here. So there's a capillary bed with these ependymal cells lining that ventricle. So in the ventricle is where cerebrospinal fluid is going to enter from the capillary through the ependymal cells to accumulate and then circulate around the brain and spinal cord. So we're talking about cerebrospinal fluid that is being produced by the choroid plexus, which again is a combination of the capillary bed and the ependymal cells. Ependymal cells are these cuboidal cells. They are ciliated with these little hair-like projections here. And so the plasma-like fluid coming from the circulatory system is going to flow through these ependymal cells, or be secreted by these ependymal cells into the ventricles. And we'll be talking a lot about brain ventricles and cerebrospinal fluid here. Another glial cell that we mentioned in the central nervous system besides astrocytes and ependymal cells are these cells here called microglia. So they are wandering cells that phagocytize cellular debris, which helps limit any sort of damage to nervous tissues. There we go. There aren't a large number of them, I suppose, because they are wandering. They're rather small and not very large in number. Oligodendrocytes, as we said, are these cells that myelinate axons in the central nervous system. And so we also talked about neurolemmacytes myelinating axons in the peripheral nervous system. So if you look back at that information, which we'll actually briefly cover again in this chapter, they surround the axons and providing an insulation here, which is going to allow nerve transmission to jump from neurofibril node to neurofibril node, which is a more efficient and faster form of transmission. All right. Oh, and one difference that you can see here between oligodendrocytes and, if you recall, neurolemmacytes is that a single oligodendrocyte can myelinate multiple axons. So you can see all these various processes reaching out to a number of axons, essentially. All right, next we're looking at glial cells in the peripheral nervous system. So there's our neurolemmacytes, which we've talked about before, which myelinate axons in the peripheral nervous system. Another glial cell that we have not talked about are these satellite cells. So satellite cells are found in supporting cell bodies in ganglia. So if we look here at this square, we're looking at, again, the spinal cord, the posterior root ganglion, and that posterior root ganglion is blown up here with these cell bodies of unipolar neurons, and these cell bodies are surrounded by these tiny satellite cells. So the satellite cells carry out a protective and supportive function for these cell bodies in the ganglia. And ganglia, as you know, are part of the peripheral nervous system, as are satellite cells. Neurolemmacytes should be familiar to you. Here we are seeing the individual, well, this is the neuron, the individual neurolemmacytes which concentrically wrap around the axon to provide a myelin sheath, or to form a myelin sheath. And one thing that I want to point out is that this outer layer, as we'll see, I'll repeat this again, is called the neurolemma. It's the outer layer of the myelin sheath. So we only find neurolemmacytes, myelinating axons, in the peripheral nervous system, which would be for within nerves, not within the spinal cord or the brain. This is a histological view of those satellite cells, so that we're not just looking at a diagram. So here again we're looking at a dorsal root ganglion. These are this whole large cell here would be the cell body of a neuron, and these little cells that the arrows are pointing at here are the satellite cells surrounding the cell body of a sensory neuron. All right, just to review myelination, we have a discussion here with, here we go, with oligodendrocytes and neurolemmacytes. This would apply to both of those. Remember that the myelin sheath is a protective, largely fatty coating that surrounds, concentrically wraps around each axon, or wraps around an axon. So oligodendrocytes are associated with the axons, myelinate axons in the central nervous system. Neurolemmacytes myelinate axons in the peripheral nervous system. And then between the cells that provide that myelination, we have neurofibril nodes, which are exposed areas of the axons that the impulse jumps between. So these slides, just remember, show how myelination occurs, how this is the case for a neurolemmacyte, how here we're looking at a cell, it has a plasma membrane, a cytoplasm, a nucleus, and it just begins to concentrically wrap around the axon. And as it does so, the cytoplasm and nucleus get pushed to the outside, while just layers of cell membrane or plasma membrane start to stack up tightly against one another, till finally we have multiple layers of plasma membrane, and all the cytoplasm and nucleus is just in this small region here. And as I mentioned earlier, this outer layer of the myelin sheath is referred to as the neurolemma, which will become important when we talk about clinical applications. All right, so we've already talked about myelinated versus unmyelinated axons and how they affect nerve impulse transmission. Just to remind you, a nerve impulse is basically a change in the electrical voltage across the membrane of an axon, which is generated by the diffusion of sodium ions through ion gates from a neuron basically to the next cell in line, which is either a neuron, a muscle cell, or a gland. So you can review the notes here on impulse transmission occurring within myelinated axons versus in unmyelinated axons, but this is where we'll pick up in the next video clip.